So the vision for the future, our mandate ought to be threefold. To improve life here, to extend life to there, and to find life beyond. NASA's mission must be driven by the science, not by some destination. We will go where the science dictates that we go, not because it's close or because it's popular or because it may be the interesting thing to do. We're going where the fundamental questions that we seek to answer take us.
You have a final range clear to launch.
In its early years, NASA focused on the science and technology that would get man to the moon and back. With that goal achieved, NASA's focus shifted to expanding its scientific research efforts to take advantage of microgravity, or the near weightlessness experienced by astronauts when in orbit around the Earth. Today, with regular space shuttle missions and the advent of the International Space Station, access to this microgravity environment is unprecedented and presents frequent research opportunities for scientists all over the world. On any given shuttle mission, there are literally dozens of payload experiments flown on board. Each of these shuttle experiments is part of 8 different research programs in NASA's Office of Biological and Physical Research. The experiments study human support in space, material sciences, the Earth and space environment, and product development. In the Fundamental Space Biology program, the focus is to use spaceflight and ground-based research to study the fundamental processes of life, such as development, biological function, and evolution. Since microgravity affects living things in very critical and specific ways, let's look at the Fundamental Biology Program experiments that are flying on the shuttle mission designated STS-107. For STS-107, NASA's Ames Research Center in California and the Kennedy Space Center in Florida have teamed up to put 6 fundamental biology experiments on board the Space Shuttle Columbia. Each of these 6 experiments is led by a different principal investigator. 3 of the principal investigators are studying the effect of microgravity on mammalian physiology, using the rat as a model. Another principal investigator is studying the effect of spaceflight on bacteria. And the remaining 2 principal investigators are studying the effects of microgravity on plants. Payload specialist astronauts flying on STS-107 have been meticulously trained to perform and maintain not only these 6 experiments, but the more than 39 experiments flying on this mission. That is a lot of training, but both the astronauts and the experiments themselves have undergone years of planning and preparation to be ready for launch. For the scientists, their journey started with a proposal to NASA, which had to pass through a rigorous review before selection for spaceflight. Then the experiments were tested repeatedly on Earth to ensure that each one has the best opportunity for success in space. In addition, each experiment must be compatible with specially designed spaceflight equipment and comply with the shuttle's limited resources of crew time, volume, weight, and power. Now, let's learn more about those 6 fundamental biology experiments. Many physiological changes take place when astronauts go into space, including adapting to microgravity, cardiovascular changes, and changes in the distribution of the body's fluids. The 3 scientists studying rats are examining the mechanisms involved in these changes so they can learn more about them. In the first experiment, Dr. Gay Holstein of the Mount Sinai School of Medicine is studying the structural reaction of the brain to a completely new environment. Specifically, she's studying the part of the cerebellum that receives balance and positional information from the inner ear.
Let's start at the beginning. You know that there are gravity sensors in the ear. And those gravity sensors are accustomed to what we call a 1G force, the gravitational force field on Earth. And those sensors detect minor changes, minor fluctuations in that force of gravity as we move our head and as we move on Earth. Those sensors send a signal into the brain, into particular groups of cells in different parts of the brain. When an astronaut is launched into space, what happens is that there's a sudden transition from this 1G force that the nervous system has developed in. So all of a sudden, that force is changed from the constant 1G to a very, very small percentage, like a millionth of that force of gravity. Space adaptation syndrome is one of the experiences that astronauts have while they're in the process of adapting to this new microgravity environment. There is also a period of readaptation when astronauts return to Earth. Both of these are considered to be processes that have a brain basis, a neural basis. I'm studying a part of the brain that's called the cerebellum, and it's located right back here. It's the small brain. And the cerebellum is best known for its importance in the timing of movements and coordination. I'm looking at this one portion of the cerebellum, that is the portion that receives gravity information from the ear. And I'm comparing the structure of the cerebellum of that from the vestibular portion, the balance portion, with another part of the cerebellum that receives no gravity information and no sensory proprioceptive information and so forth, which presumably should not be affected by a microgravity experience. My question is, do I see a difference in the architecture of the portion of the cerebellum that receives gravity information in experimental animals that have been in microgravity and have just returned? So I'm looking for the structural basis for this reorganization in the brain that occurs during the readaptation process.
The next experiment is being conducted by Dr. Michael Delp of Texas A&M University. Dr. Delp is studying the specifics of how the cardiovascular system changes during spaceflight.
The basis of our experiment is really to understand the effects of microgravity on the cardiovascular system. The body is designed to adapt like a weightlifter or an athlete adapts to training. They become stronger or faster. In microgravity, the body also adapts. And when astronauts come back to Earth after being in microgravity for a relatively short period of time, many of them are unable to stand, and it's because they cannot maintain an adequate blood pressure. And so the purpose of our study is to really try and understand why do the regulatory processes that govern blood pressure, why are they altered in microgravity? And those involve the heart, the veins, and the arteries within the body. One of the properties of arteries and veins is that the wall is composed primarily of smooth muscle cells, and that's how these blood vessels constrict, and then they dilate by the smooth muscle cells relaxing. And in microgravity, we believe that these smooth muscle cells actually atrophy because they're not used as much, just like skeletal muscle. And so when the astronauts return to Earth, these muscle cells are not as strong. They cannot constrict the arteries or the veins as well. And so there are problems with blood pooling and also with regulation of blood pressure. And so when the astronauts come back, they stand in an upright position, their blood pressure is abnormally low, and so therefore there's not enough blood going to the brain. So we want to understand exactly what is happening to these arteries, veins, and then also the heart, whether the heart is getting smaller in microgravity. So whether microgravity alters the structure and the function of the arteries and veins has virtually never been looked at. So we're very excited to see whether or what is happening, and those are really the focus of our studies.
Dr. Jacqueline Gabrion of the Université Pierre and Marie Curie in Paris is studying the regulatory mechanisms which control the production of cerebrospinal fluid in microgravity.
J'étudie depuis plusieurs années les régulations de...
For the last several years, I have studied the body's production of cerebrospinal fluid, the liquid that surrounds our brain and is secreted by a small organ called the choroid plexus. The choroid plexus is very sensitive to physiological and environmental conditions. During previous shuttle missions, we were able to demonstrate that microgravity affected the ability of the choroid plexus to produce cerebrospinal fluid. Following these first experiments, we decided to continue studying the control of this production. This experiment entails studying rats to obtain clues on particular proteins which are called aquaporins, the channels that transfer water from one compartment to another through biological membranes. These proteins are very important for the brain, but also for the kidney and the lungs. We plan to study not only these mechanisms, but also the neurotransmitter receptors which are expressed in the choroid plexus. Currently, we are quite confident that new results will confirm our previous findings, which are that adaptation to microgravity triggers a reduction of the cerebrospinal fluid production. And this reduction is controlled by the aquaporins proteins that transport water and by the neurohormonal regulations which affect the choroid plexus. With the help of this kind of information, I believe we will be better able to understand the fundamental function and fundamental physiology of this organ, and also get a better handle on how it adapts to the extreme conditions of spaceflight.
All three of these rat experiments will help give scientists a greater understanding of fundamental biological processes and provide important clues as to how microgravity might also affect the human body. Our next principal investigator, Dr. Barry Pyle of Montana State University in Bozeman, is studying the effects of microgravity on a common species of waterborne bacteria.
Our experiment is based on some earlier observations with bacteria in microgravity where they were found to grow faster and sooner than the ones that were grown on Earth. And we're looking at not only their growth but also their ability to cause infections. We hope to learn more about the growth of bacteria in microgravity, largely because in space it's also been shown that the crews may become more susceptible to infections, and also bacteria may become more resistant to some antibiotics, which means that it may be more difficult to treat infections in space. Our experiment entails putting the bacteria in a suspension of water in some small chambers in our flight hardware, and then on about the 8th day of flight the crew will inject these into some culture media which is in another chamber in the hardware. They'll incubate them for 24 hours at body temperature, at which time they will end the experiment and put them back into storage in a canister that keeps them at refrigeration temperature until the shuttle lands. This experiment is actually funded by NASA, and we're using European Space Agency equipment because they have the sort of equipment that we need to use, and also they have an incubation system that they will be flying on this particular shuttle mission. It's exciting to work on a spaceflight experiment. There's a lot of details to be completed, and to make sure that everything works properly. We only get one chance to do this, and so we have to make sure that everything goes right. And actually having students and others working in the laboratory and getting them excited about spaceflight and space science is really interesting for me too.
The results of Dr. Pyle's experiment will give us a broader insight into how bacteria that may affect humans responds to the microgravity environment. The remaining two fundamental biology experiments on STS-107 involve plants. Dr. Carl Hasenstein of the University of Louisiana, Lafayette, is testing the gravity-sensing mechanism in flax seeds.
Well, the basis for the experiment is to hopefully figure out the true gravity sensing mechanism in plants, and more specifically in roots. There are a lot of different models out there that all attempt to explain how plants sense and respond to gravity. And one of these theories is based on the displacement of dense particles, so-called statoliths, within sensitive cells, statocytes they are called. And what we try to accomplish with this experiment is to take away gravity by going in orbit and substitute a directional force by so-called high gradient magnetic fields. And when we can hopefully succeed in displacing these amyloplasts because of their magnetic properties and induce curvature at the same time, we would have probably one of the strongest pieces of evidence to support this statolith-based gravity sensing model. The experiment is important because we hope to understand and be able to answer the basic question in biology: how plants perceive and respond to gravity. So it can go in different directions. We can understand a great deal about the so-called signal transduction chain that controls plant growth in general. But it also has an applied side where we can hopefully determine ways to allow plants to become oriented in microgravity or in reduced levels of gravity. So when gravity is not sufficient, as in space flights, we can substitute this or replace it by these high gradient magnetic fields. And this would allow, at least initially, to determine the growth axis of a germinating seedling. And that's an accomplishment that will hopefully help future plants to grow plants in microgravity.
And Dr. Fred Sack of The Ohio State University is conducting an experiment on how gravity-sensitive moss cells develop in microgravity. Dr. Sack's first version of this experiment was flown on an earlier shuttle mission, STS-107, where the moss responded to spaceflight in a completely unexpected way.
We're looking at a moss cell that grows away from gravity, and we're also trying to understand how these cells sense that gravity, and furthermore trying to see the forces that act within these moss cells that helps gravity shape cells themselves. So it's one thing to shape an entire tree through the wood and through the shoots growing up, but what about all those individual cells that make up those trees? And so we're looking at both the response of these moss cells to gravity to figure out how they sense gravity, what they do in the absence of gravity in space, and also to get an idea of the mechanical forces that are within those cells that control the distribution of heavy organelles. Quite frankly, for STS-107, we were just wondering what would happen. It was almost as if, is it going to be randomly oriented in space, or is there going to be some other pattern? Much to our surprise, when we opened the locker and the dishes, we found that the moss grew in spirals. So the older cultures actually grew, so the tips of the filaments were circling in a spiral. Moreover, they were growing in a clockwise spiral. The fact that it grows in clockwise spiral says that in space the growth is non-random. And this is a unique finding for most of spaceflight experiments, because in other plant systems the roots or the shoots would grow randomly. So the fact that we were uncovering a non-random growth pattern in the absence of gravity is really quite novel, and it suggests that there are some underlying endogenous forces that are present and acting in the dark, but normally gravity just overrides them and we don't see them.
So what do you think is the next step?
I think that learning about how cells work and the role that gravity plays in that could have spin-offs, because that's how science advances. People uncover some unusual phenomena through a specialized system, and then they learn something that makes them look at other systems differently. And that can lead to discoveries that can have impacts down the road. So fundamental biology is intimately tied with spin-offs, but sometimes it's hard to see how. And you just have to follow the questions to see where it leads.
The results of both of these plant experiments will expand our fundamental knowledge of how plants sense gravity and also the basic mechanisms of cells themselves. So now you have an overview of the fundamental biology experiments which are flying on STS-107. The principal investigators and their teams of researchers will be studying the results of these experiments long after the shuttle has landed. And the findings that they glean from these experiments will lead to more questions, more experiments, and more findings. And ultimately, this will lead to a greater understanding of the human body and the world we live in, which is pretty much what NASA's mission is all about.
Frankly, the thing I find most exciting about this experiment is how humbling it is to do an experiment on the shuttle. To just watch an experiment that you've prepared and it worked industriously on, and it just occupies one small piece of a mid-deck locker, and then to watch this massive machine take off is breathtaking. And while we're very excited about the work we're doing, and we think it's fun, full of questions, it all gets put into a kind of perspective when you see the machine that's carrying it, and it gets a new seriousness, and it gets part of a larger awesome whole when you see that shuttle take off and land.